Now that we've covered transcription and DNA replication, prior to that, we're going to look at translation now, the genetic code, and ribosomes. We've seen this figure before. Again, those little orange mousy-like figures are the ribosomes on which protein synthesis takes place. The orange shapes, as you can see, there are two kind of held together here, represent the two components of the ribosomal complex. The little tails are the nascent protein chains that are being synthesized. The linear strands, 5' to 3', between the ribosomal subunits represent the mRNA. So here we have the mRNA, and we're looking at the prokaryotic side here. We can't see the tRNAs, but they're there doing their duty. They're just too small. And again, to remind you that there is between prokaryotic and eukaryotic translation, there is spatial separation in eukaryotic cells, transcription, replication, transcription occurring in the nucleus. mRNAs are transported out to the cytoplasm where translation occurs on the ribosomes. This can be concurrent in prokaryotic cells, so they can occur at the same time. Several types of RNA play key roles in translation, and here is a table that indicates what those are. You're not responsible for any numbers. Any numbers that are given are just with respect to mass and number of nucleotides are just to reflect the size. So we have ribosomal RNA, transfer RNAs, and mRNAs. So we have components of the ribosome, transfer RNA components, and that messenger RNA. So in bacteria, an mRNA may code for a single gene, but more often it codes for a cluster of genes. And this is called polycistronic. So a piece of mRNA that is going to be translated into more than one protein. Also, and I keep forgetting to pick up my pointer here, in eukaryotic cells, a distinct mRNA is produced for each gene and thus one protein. Those are called monocistronic. Okay. So polycistronic versus monocistronic. There are a number of features of the genetic code that we should take a look at. A sequence of 3 bases in mRNA carries the code for one amino acid in the final protein product. The genetic code is non-overlapping, which means that given a stretch of mRNA has only one reading frame, or given a stretch of an mRNA, there is only one reading frame, which only codes for one protein. The mRNA is then translated 3 bases at a time from a fixed point to yield a single defined product. The genetic code is commaless, no commas, meaning the codons are read continuously with no spaces in between them. The code is universal. All organisms use the same code, although there are minor exceptions which we won't discuss. The code is also considered to be degenerate, indicating that several different codons specify the same amino acid. The use of 4 different bases to form 3 base codons gives 64 different combinations. Because there are 64 ways to code for 20 amino acids, the degeneracy of the code is not surprising. again, the message will be read 5' to 3'. So there are reading frames within the mRNA that can be read only one way. So the reading frame describes the requirement for a specific start site on the mRNA, which results in the reading of a distinct set of triplet codons. Because the translation uses triplet codons, there are 3 possible ways in which a series of nucleotides could be translated. However, there is only one correct reading frame, which codes for a specific functional protein, and 2 incorrect reading frames. And I thought I had put these all, oh, here we go. Here it is on one slide. So here we have the reading frame. Okay. If we shift the triplets 1 frame shift, we get a sentence that has words that mean nothing to you and I. If there is a 2nd frame shift, we still have that incoherent set of triplets that do not code for an amino acid. So frame shift 3 gets us back to the original message. The big dog saw the red fox eat the fat cat. So if the first codon should be skipped or frame shift, well, if the first codon is skipped, we would eventually get back to the correct codon. However, if we have a frame shift, we will get nonsense. The whole point. So this is a table of the 64 codons. 3 nonsense or 3 stop codons, which code for the termination of translation, are present. Here we have a stop, stop. This is also a stop. This is a start. Okay. So these are considered to be nonsense or stop codons. You should know the sequences of these codons, the stop codons, and you should also know the sequence of the start codon that codes for methionine. And it is usually the first amino acid to be incorporated into the protein. So here we have a matrix that only shows the amino acids, and this is to help us see a few important features. The stop codons are clustered together, is one feature. The only amino acids encoded by a single triplet are methionine. And where's tryptophan? Trying to find it. Where's TRP? And all this, I can't even find it. But tryptophan is the other amino acid that only has one single codon. Can't believe it's not jumping out at me. Oh, here it is. Here we go. So single codons for methionine and tryptophan. If you will notice that the amino acid for which there are multiple codons are generally clustered in a single box. It is often the 3rd nucleotide of that triplet that differs for a single amino acid. And you can actually look at that on the previous slide as well. This clustering, though, makes it more likely that a point mutation that changes only one base in a triplet will not cause a change in that amino acid sequence or translation to the appropriate amino acid. Note as well that similar amino acids are clustered together, polar and nonpolar. So in this box we have polar, 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 and we've got amino acids that are, I'm sorry, I should have said nonpolar. These are polar amino acids, so they tend to be clustered together. So again, if a point mutation exists in one of those three bases in the codon, that substitution will be a similar amino acid, and the effects of that substitution will be minimized. What we're looking at here is a nucleotide sequence of a set of genes coding for a protein called capsular polysaccharide. We have the coding strand of DNA. We have the open reading frames. So the DNA that codes for this protein is about 2,500 nucleotides, and the corresponding amino acids are also given. Note here we have that promoter sequence in the DNA at -35, -10. We have the +1, our start of transcription, and then we have translation beginning here. We're going to focus on a small amount of this region in the upcoming slide. I believe it is. Yeah. This is magnified from the sequence in line 3. And the typical features of the 5' end of an operon being the unit of genomic DNA containing a cluster of genes under the control of a single promoter. So this would be a prokaryotic promoter. So this would be a polycistronic situation. The genes are transcribed together into an mRNA strand and either translated together in the cytoplasm or undergo transplicing to create individual mRNAs that are translated separately. So the DNA here contains the upstream region where the regulation of translation takes place, as I mentioned prior. So here's our -35, and here's that -10, the Pribno box. The other sequence that is, and here we have the start of translation is always a sequence of ATG. This is the A from that transcriptional start site, and this ATG codes for methionine. The other element that is present here is what's called the Shine-Dalgarno sequence. And this is the binding site for the 16S ribosomal RNA, which indicates where that ribosome needs to start thinking about translation.